Welcome back. So what I'd like to talk about now is how new sections of STEM with those nodes and internodes are going to get made. And I want to start with kind of just this overview. So over here, we can see this diagram of a plant. Below this imaginary line, this would be our root system. And you know that there's a main root. At the tip of that main root, there's a root apical meristem. That's where new cells are going to be produced, where the primary meristems are going to get produced, and eventually our primary tissues are going to get produced. Well, that same process of primary growth or elongation is also going to be going on in the tips of our lateral roots or branch roots. Well, today we're going to be covering the process of growth that's occurring in the shoot, the above ground portion of the stem. And if we look at our shoot here, at the very tip, there's that terminal bud, that shoot tip. That's going to be going through this primary growth process, elongating, forming new sections of stem. If we look at this node right here, where this leaf attaches, here, that axillary bud has already grown into a little branch, right, that has its own nodes and internodes, and a terminal bud right here, and those axillary buds all along. So we're going to be drawing this process as it happens, and I'm just going to diagram a little shoot tip. We're going to be diagramming shoot tip anatomy to visualize this process of primary growth or elongation, whereby our stem is elongating and we're getting new little leaves produced. And so what I'm going to draw is I'm going to kind of center my drawing here. To me, these always kind of look like little monsters. All right, this is my little shoot tip. And ultimately, we can trace all of the cells in the shoot back to our shoot apical meristem, or SAM. So below ground, we got the RAM, the root apical meristem. Above ground, we've got the SAM. And it's literally just sort of a dome of cells right here. And so that's our shoot apical meristem. That's where we're going to get lots and lots of mitosis to produce our developing leaves and to produce our developing stem. Now looking at this little monster, these big little flaps that I have drawn here out to the side, each one of those is a developing leaf. And since they're young and underdeveloped, I'm going to call them leaf primordia or leaf primordium singular. You may have heard of like the primordial soup, referring to like the beginnings of life and the beginning of things. This just means beginning underdeveloped, meaning that these cells haven't turned into epidermal ground and vascular tissue yet. So those are our leaf primordia. You can see they're developed externally on our little shoot tip. You know about leaves already. You know that all leaves are going to have axillary buds. So inside those axils are going to be axillary bud primordia. Maybe I'll label this one. And I'm just using that word primordium to indicate that they're young and underdeveloped. So we can see those externally on our little shoot tip. Well, everything that we're seeing here is sort of young and underdeveloped, hasn't turned into primary tissues yet. And what we're going to see is that our shoot apical meristem is going to produce the same primary meristems that we saw in the root. So we're still going to see the protoderm, procambium, and ground meristem, right? Those same primary meristems you've already seen in the root, but their arrangement is going to be a little bit different because we're above ground now. Well, let's start with that protoderm. So the protoderm is going to be just a single cell layer on the outside of our developing leaves and our developing stem. And just like it did in the root, that's going to develop into the epidermis of, in this case, our leaves and our stem. Well, running in strands is where we're going to find our procambium. So it's going to be running in strands through our developing leaves, through our developing sections of stem. So in red there, I've just indicated the procambium. Just like it did in the root system, that procambium is going to turn into the vascular tissue of the plant, right? In this case, of the stem and the leaves. Well, as far as our primary meristems go, everything that's not procambium and that's not protoderm is going to be the ground meristem. So all of these other intervening cells out here and here and here, everything in our leaf primordia that's not either protoderm, just that single cell layer on the outside, or these strands of procambium, it's just going to be that ground meristem. So all of that is going to be turning into the ground tissue of our shoot system. Well, let's look at this briefly on my PowerPoint. All right, here we see a close-up of that shoot apical meristem, or SAM. Let's get a bigger view of this. So here's that little shoot tip, right? Similar to my drawing here. That shoot apical meristem right here. You can see the just very beginnings of these leaf primordia here. Here's bigger ones. Here's bigger ones. You can see these little axillary bud primordia, right, in our leaf axils. You can see these strands of procambium running through those developing leaves and running through our stem right here, our developing stem, and right here. Let's see, our protoderm, just a single cell layer all along the outside. And everything that's not the protoderm is going to be the ground meristem. So all of this out here, right over here, right over here is ground meristem. Now, thinking about how our leaves and our axillary buds originate, they're going to originate externally and enlarge as they go through this process. You can see them here just starting these little nubbins. Here they look more like horns. And here they actually look like fully developed leaf primordia. Now, one kind of cool thing, right? Remember, the protoderm is just a single cell layer all along the outside. Ooh, what do you think these funny things are coming off of our epidermis here? Those are trichomes, right? So we can see trichomes, or these epidermal hairs, developing along that outer edge. Well, the last thing I'd like to do before we're done with this for today is I want to imagine a section of stem that's down further, right? That's down further, where all of our primary meristems, right, our protoderm, our procambium, and ground meristem have turned into primary tissues. And what I'm going to make is I'm going to make a drawing of a eudicot stem cross-section. Cross-section, remember, I'm abbreviating just like that. This is going to be a eudicots stem cross-section showing those primary tissues. Showing where all of those primary meristems have already turned into our primary tissues. So we're going to start on the outside and work our way in. So I'm just going to draw a circle. As most stems, you know, kind of circular. On the outside of our eudicots stem cross-section, we're going to find our epidermis. That's just going to form from our protoderm. Just a single cell layer. We might find some trichomes there. We're going to find guard cells and stomata in that epidermal layer. Well, in a eudicots stem, our procambium is going to turn into vascular bundles. And in a eudicots stem, we're going to see that our vascular bundles are arranged in a ring. And I'm just going to draw a few vascular bundles. There really are more than this in reality. When I make these models, I simplify them so we can talk about things, but reality is always more complicated. So each one of these is just a vascular bundle. Each one is matured from a strand of that procambium. Well, remember, whenever we see our vascular tissue, we're going to find those two parts, those two components. We're going to find our xylem and our phloem. Well, our primary xylem, remember, we're going to keep on diagramming in red. The reason I'm doing that is just to make patterns, things more easier for you to see. That's going to form the inside of each vascular bundle, closer to the center of our stem. So that represents our primary xylem right there. Doing exactly what you think it's doing, right? It's going to be transporting water, right, from the roots up on the plant. So that's the inside of our vascular bundles. The outside of our vascular bundles are going to be primary phloem. Remember, I'm going to keep drawing that in blue. That's going to be the outside of each one of these. That's going to be doing exactly what you think it's going to be doing. It's going to be transporting sugars from photosynthesis from wherever they're produced to wherever they're needed or wherever they're going to be stored inside the plant body. All right, so that's our vascular bundles, right, forming a ring overall in our eudicot stem cross-section. Well, the last of our tissues that we need is our ground tissue. Well, because our vascular bundles are in a ring, we can differentiate different regions of ground tissue based upon the relationship to those vascular bundles. Now, if I'm in this region right here, right, in between our vascular bundle and the epidermis, we're in what we call the cortex. We use this term in the root, right, to refer to a similar region. If I'm inside of my bundles of vascular tissue, we're going to be in the region of ground tissue called the pith. We use that term in a root too, right, to refer to ground tissue that was inside of vascular tissue. Well, I'm going to give you one more, and that's this region in between our bundles. These we call pith rays. These are just rays of pith that radiate out in between our bundles of vascular tissue. But all three of these, cortex, pith, and pith rays, all of these are just ground tissue. And all of these are going to form from the ground meristem as it matures into these different regions. Well, in lab this week, we'll be seeing eudicot stem cross sections. We'll also be seeing monocots as well, but I'm going to leave the details of the monocot stem structure for us in lab. I'll see you soon.